All right, back at it again, crew. Um, my camera angle's a little different here. I got a new setup, so that's why I'm a little delayed in between videos. All right, so today is going to be, I think, kind of a shorter video. We're going to talk about loading data sets. So up until today, we've really just created data sets in R. But what happens if you have like an Excel sheet or some sort of spreadsheet with your data in it and you want it to get into R so you're able to use that data because you don't want to have to type everything in like we've been doing before. So we are going to look at how we load it. So first thing we're going to have to do though is get data, right? So we're going to go to this webpage. It is NCBI, and they have a resource called DBGAP, which is database for genotypes and phenotypes. And we are going to then click on the phenotype genotype integrator right here. And so we're just going to get some data just to kind of show basically how we load it up. So you can do this with any CSV or Excel file. But I'm going to click Browse here. And we're going to pick some, where are they? Let's do some mental disorders. And so this database is a phenotype genotype integrator. And so what it does is it takes genome-wide association study data, which is data where we've taken SNPs or molecular markers from across the genome, and we've correlated those with the prevalence of some disease. And so this is very popular or very widely used in human genetics because we're not able to do things like crosses. Like you can't take a person that's got a disease and a person that doesn't have the disease and say, hey, you two have a baby and we'll see how the baby turns out. So instead, what we do is we can take a very, very big data set, so tons and tons of individuals, thousands usually, and look and characterize every SNP in their genome, and then see if that SNP is unique to people that have a certain disease. And if that's the case, then we can assume that SNP is in the neighborhood of this disease. That's a very basic way of putting it because this all recombination comes into play here. So you have SNPs nearby a gene that aren't necessarily within the causal gene itself. That's why I say you can find kind of the neighborhood of the causal gene. So I'm not. This could be a whole lecture series getting into GWAS, but it's a very powerful tool. It's really cool. All right, let's pick a disease here. So we're going to pick Alzheimer's and hit apply. I guess you're supposed to add traits, then apply, but it'll automatically add it for me. And we're just going to do a single chromosome. So let's just pick the first chromosome. And so we're going to look for SNPs related to Alzheimer's disease on that first chromosome. We're going to click search. And so this is a database, or the results from the database. We have our Alzheimer's disease here. We have whether or not the SNP was an intron within the gene, a missense, a 3' untranslated region, 5' untranslated region, etc. We have the gene that's associated with the SNP, the location, so chromosome 1, and then the base pair number, the p-value of how significantly associated it is with that SNP, the source of the experiment. They don't have anything for study here, but the population. So we can say, this was done within a European population, African American population. That seems to be it for Alzheimer's. And then the reference to PubMed. So what we're going to do now is we're going to click download. And I'm just going to save it to my documents folder here. And I've already saved it once practicing or trying this out. And so I'm going to save it as alzheimers.tab and click save. And for me, I'm going to replace it. And now we're going to go back and we're going to do the same thing 2 more times. So we're going to pick 2 more mental disorders. So the other one I picked before was dementia. Add that trait and apply. We're still going to do chromosome 1. Going to search and see we only have 4 SNPs here that are related to dementia that have been identified. We're going to click download. I'm going to save this one as dementia .tab. And then we're going to go back and we're going to pick 1 more. Remember, this is just trial stuff, so I can give you a demonstration of how to load data. So you obviously could load your own data as well. And I think the last one I picked was bipolar before, so we'll click apply. Again, we'll stay with chromosome 1. We don't want examples. Let's search. And here we have about 50 SNPs that are associated with bipolar. So we're going to click download. I'm going to save this one. Change the names of how you save them because they're all going to save as a phi gene association, so phenotype genotype association. What was this one again? Bipolar. So we'll say this is bipolar. Replace it. Okay, now we're done here. So you can get out of the database. But this is a cool database to look around in if you want to do some stats or practice graphing and stuff like that, because it's a bunch of cool free data to access. Okay, back to R now. So the first thing we're going to do, we're going to use our Markdown file that we've started previously to just kind of keep updating this as we go through the course. And this way you'll kind of build your own textbook. So I encourage you to make sure you annotate it well, even better than I annotate mine. I kind of take shortcuts there. So here we're going to say, example, we are going to learn how to upload our own data. Okay. So as we talked about previously, there's all these packages, that R has built in them or people have written before. And so we're going to actually use some of their packages. And so we're going to do first. So what you're going to have to do first is you can just go up to install packages up here. And we're going to do read bulk. And we're going to click install. It might ask you to restart R. That's fine. And I already had it installed, so it's going to be fast, so it might be a little slower for you. And then the other one we're going to install for this example is Plyr, P-L-Y-R. Install that. This one might take a little bit of time. I think it's a pretty big package. Okay, and so now that we've installed them, we're good to go. But we need to call them. So whenever we install a package, it goes into like our behind the scenes of our studio, but they're not always active. And so what we have to do is type in library, and we're going to do library read bulk. There we go. And then we're also going to do library Plyr. And I'm just going to leave this chunk by itself. So I'm going to say, first we load the libraries. You can integrate the install into it, into your textbook here. However, you might want to put eval equals false after you've installed it the first time, because you don't need to reinstall it every time, and it might slow down your code. So we get a little warning here that read bulk was built for a different R version. Both of these were. That's fine. But if you have errors that come up later, this lets you know. So this might be a problem for it. But I don't think we'll have that problem today. All right, so I'm going to make another code chunk here. And I'm going to first let's load data sets individually. I'm actually going to clear this out because this was from making sure my code worked. All right, so let's do Alzheimer's. And so we're going to load it and store it into a variable. And what we're going to do is use a command called read delim. So this reads a file in table format and creates a data frame from it, which corresponds to the lines and variables to fields in the file. So that's exactly what we want to do. There are other ones as well. There is, I think, read.table. If you type read here, it'll kind of show you them all. So you can read a CSV. So if you have a comma separated value file, CSV2, delim is what we're using, table, there's a whole bunch of different options. We're only going to go through one, but if you have data in a different style or different, saved as something different, just be aware that there's a ton of different options for reading your data. So there's always going to be a way to get your data in there. So all right, read delim. We're going to close or open up our arguments here, and we're going to do Alzheimer's.tab, and we're going to say for our argument of header that it does have a header, because we did look at it previously. Let me pull it up here real quick. Documents. So you might want to check this. Oh, it's going to let me pick. Ugh. Let's see what it looks like in Notepad. Perfect. Okay, that'll work. So you could open it up in Excel and it would be nicer, but I didn't have the option. It went search for it. But we do see that we have headers up here, right? So we do want to say header is true. That way it won't include that in the actual data. And lastly, we're going to tell it what kind of separation there is between each column. If it's CSV, it's going to be a comma. But in our case here, we are doing /T, which means a tab. So the way it came from that database was tab delimited. So that's what we have to use. But if we would do this question mark read.delim, let's run this line. As you can see here, if we go down to SEP, you can have white spaces, tabs, new lines, returns, etc. So you can specify that to whatever your data set is. Okay, and so we're going to do the same thing. I'm just going to copy and paste, but we're going to change our variables to dementia. And we're going to change our file to dementia. All right, so now we're going to run both of these. Perfect. And so up here, we can click on it, and you see that we have our 4 genes for dementia. Our headers are actual headers, and they're not a row of data. So if I take this off, let's run this again real quick with headers equals false. And so you see now this is actually in a row of the data, and our headers are just numbers, which we don't want. So that's where like the header versus true versus header versus false comes in. So let me run these both again. You can see we got our 4 dementia genes here, intron, intergenic, what the gene is, etc. We look at Alzheimer's, we got 63 here. Ooh, there we go. Intron, blah, 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 blah. So perfect, that's what we wanted to do. So now though, what if you have, such as the case of my lab, when I am, my students in my lab are generating an Excel spreadsheet for every individual plant that we're looking at. And we're looking, in the long run, it's going to be like a couple thousand plants. I don't want to write a couple thousand lines of code to load every single individual plant into my data set, right? So that's where this package read bulk comes in. And so if I do this double question mark, because it's kind of an external package, oops, we probably need to put that within an R script, don't we? And I'm going to close it off. All right, so if I type read bulk, Um, this is a way to read and combine multiple data files without having to type out every single one. All right. And so I'm going to get rid of this, but you can keep that in there. But we need to annotate this. Let's load multiple spreadsheets into a single data frame. So this will save you a lot of time if you've got a lot of data, which R is great with a lot of data. So we're going to call these mental disorders, right? That's where we got our data set. And we're going to use read bulk as our actual command here. And now we're going to tell it the directory because it has to know where to look. And by default, oh, this reminds me, one thing to take into account is you need to save these files to the same place that you're running your R script from. And so you can change that by going to, where is it up here? Session, set working directory, and you can set it to the source file location. So that is where we have saved our markdown file, or you can choose your directory, but R needs to have, in this case here, needs to know where the file is. If you have nothing here in front of your file name, then it's just going to assume it's in the directory that the R markdown file is. You can put like C:, actually, and then like my computer and put the whole length to the file. But I've just found it easier to put the data in the same folder as the R markdown file or R file. Okay, back to it. So for me, I am using, this is going to let me copy and paste. This is where I stored mine in. So users, funny story, when I got my new Surface, I don't know why, all my other Surfaces had Josh as my username, but this one assigned me the G, and I've never changed it. I don't think I'm a G. I don't know where that came from. But then within my OneDrive and documents, so that's where I saved my R file to, and that's where I saved my those tab files to. Okay, so now we have to, for the read bulk, we have to tell it what function we need to use, and this is that big list that I showed you before, like read tab, read CSV, read delim. So you can choose which one of those you want this bulk reading to use. So we're just going to use the same thing. We're going to use .read delim. And then I think that is it. Let's try it. Cannot open the connection. Did I not put them in that folder? Documents. Okay, so we're going to troubleshoot why it's not picking them up. So properties. Users, the G, OneDrive, documents. Oh, I know what I forgot. So the reason it's doing it is because I have to also tell it what extension that I want to be used. So because there's all like weird files in there beyond our tab files, I need to tell it only open tab files, don't open PDFs and stuff, because it's not going to work. It's not going to like that. Where did my read bulk? So the argument is extension. So I'm going to say extension equals, and I'm going to put .tab. And now let's try it. Okay, so it says reading Alzheimer's.tab, reading bipolar.tab, and reading dementia. Those were the three that we had. Ignored everything else that was not a .tab. And we go look at our mental disorders here. And now we have Alzheimer's, followed by bipolar, followed by, oh, why it does this weird glitchy thing, followed by dementia, all in a single data set. And so, wonderful. If this works for 3 files that we use today, it works for 1,000 files. If you have it in there, it might take a little bit of time to run through 1,000 files. But as you can see, that's a lot faster than doing individual ones. And we'd have to merge these. We're not going to do that today because this read bulk did it for us. But you could do that if you wanted to. So now let's just do, go down here. And so just to kind of polish it off, it's kind of no fun to read files without doing anything. We're going to do a simple calculation using our data sheet that we made. So we'll do count, we'll do files. So this is just going to count, oh, actually, not files, mental disorders. So as you would guess, this function just counts things, and we're going to tell that the variable is population. So if we run this, and now we look at population, we can see the number of studies. It just counts and puts them into bins, right? So of our, how many lines do we have? 130 different lines or observations. 13 of those were from African American populations. Actually, it's more than that because, so this is another good point with our, when you're getting data. Sometimes if you get data from different sources, they annotate it differently. Oh, I guess, no, this is a different. So this is African or African American, and this is just African American. Then we have Asian, African, European, African American. So it's just a hodgepodge of a lot of ethnicities. And then we have an East Asian, Asian European, etc. And then NR not recorded. So 32 of those were from that. So we're going to cut it there. What am I at? 22 minutes. Oh, a lot longer than I thought. I said it would be a short one. But that's how you load data. And from now we can actually work with some more genetics type stuff going forward. So if you like the video, give me a thumbs up. If you didn't like the video, hit thumbs up 2 times. Hit thumbs down 2 times just to show me how much you didn't like it. And I'll see you in the next video.